So welcome back to our neurophysiology introduction. So I want us to explore the basic physiological activity of our neural cell. How do we generate impulse? Even though impulse generation for now will not be as clear, but we'll just have maybe one of the illustration and maybe the propagation. That is what maybe we'll be detailing, and also in terms of neurotransmitter release generally in general sense. So we've said that for a neuron, we've said that a nerve system is a communication or a communicative unit, a control unit. So that means there is need for an information to be relayed. So information relayed in the nervous system is normally relayed in nerve impulse. So there's some form of electrical impulse that is normally relayed. So an impulse in the nervous system is likely to be initiated by the sensory nerve endings, as we are able to see in our introductory mapping, where we had the sensory going into the spinal cord, brain, and eventually back to the peripheral using the motor wing. So the initial generation of impulses ought to be from the sensory nerve endings, and we'll be able to see not all information will entirely be initiated in the sensory area. So we'll be able to see that sometimes the brain or the CNS can automatically generate some impulses. So in these instances, some impulse, of course, maybe are possibly unwanted, uncoordinated. They are just being generated. They are not serving any sensory role. They are not going to control any initiative that was started by the sensory nerve. So these are likely to give us an coordinated nerve impulse generation that can grossly or can be clinically seen as maybe some form of convulsions. So it's also good to note that. But in a physiological state, you anticipate the neuron generation to start with the sensory nerve. So let's just explore what happens. So we are having that text. The transmission, of course, of the impulse occurs across the plasma membrane or the cell membrane. At the resting membrane, the potential of the membrane is polarized due to the difference in the concentration of ion. So this is brought about by the sodium-potassium ATPase in the state of resting. So this is just like what we did in the cardiovascular system. So we are assuming that when there's no any impulse being generated, the cell is obviously resting. So a resting cell, how does it normally look? So we said that a resting cell, of course, has channels. So we just choose any spot as channels. So we want to respect major two ions. So possibly the potassium ions and the sodium ions. And on this other side, it also has a pump, an enzyme related pump. So that is a sodium. So let's write sodium, a potassium, ATPase, able to break down the ATP. The ATP is used in as an energy that allows for the movement of this ion. So mostly we normally have the ion, or rather the sodium, being extruded out in exchange for potassium that is normally incorporated. So this is what happens ordinarily when the cell is resting. So as a result of that, you are likely to have less number of sodium inside. So the sodium, so let's write maybe sodium and write the difference below. This is not standard, but let's just use this. And as a result of this, because we are losing a lot of sodium by this active transport, so the cell is likely to have low number of sodium. Oppositely, what is happening to the potassium? because the cell is actually incorporating a lot of potassium, it is likely to have more potassium on the inside, and this compares to the extracellular area. So as a result of that, still in the resting phase, ordinarily what normally happens, you'll be expecting some form of diffusion. Now respecting the simple concept that substance or ions are likely to move from a region of high concentration to that of low concentration. So what movement will you anticipate with the potassium? So the potassium is likely to get out of the cell using now the diffusion process. Because there is a specific channel that need to be used, you'll refer this as a facilitated diffusion using some potassium channel. And because we are having less sodium on the inside than on the outside, definitely sodium will need to diffuse in. And remember, the diffusion gradient has been caused by the sodium potassium ATPase. Again, by the same facilitated diffusion. So this would normally represent the resting phase, or you can refer this as resting potential. So you can assume this as any point of the cell membrane or any point of the cell within the nervous system, or rather within the neuron. So when an action potential comes, there is normally this will shift from a resting into an action potential. So how is the impulse normally generated? So let's assume that this is let's say a nerve ending, the cell of a nerve ending, giving us a sensory modality such as something like, let's say, stretch. Stretch is reasonable. So when there's some form of stretch, let's say the skin is being stretched, the skin is will take this impulse eventually up to the brain, and then now, depending on the intensity of the stretch, it could be interpreted just like a normal stretch, or sometimes even pain, depending on the intensity of the stretch. And then of course we'll be having various motor feedback. But now on to the impulse generation. So when you're having some form of stretch, so that means our nerve ending is interacting with the now environment. It is banking on the stimuli to initiate the action potential. So what it happens, so maybe the stretch will have some form of change in terms of the flow of the potassium, in terms of the flow of the sodium. So the sense modality can be able to make some conformational change that will allow the sodium gates to open. So maybe if you use that word directly, you can assume that it will stretch the sodium gate so that it allows more sodium to enter. And at the same time, while it's stretching, it is somehow closing the potassium so that less potassium will be lost to the outside. So as a result of that, it means that with the accumulation of sodium inside and retention of potassium inside, our cell is likely to be more positive because we are retaining a lot of cation. So this represents an action potential. And remember, we are assuming we are having a cell, and this is happening at a single point of a cell or in a membrane. So once there's an action potential in this side of a membrane, so it normally spreads. So it normally spread towards the axonal end. So action potential basically meaning our cell is becoming more positive as a result of the movement of the ions or the cations that we've been able to propose. So we are, if you can animate it a little bit. So let's assume every other place was resting, we've had an action potential, so it will just spread from this point, this point, this point, this point, up to the synaptic knob, where we are having storage of neurotransmitters. And remember, every time this becomes positive, or when the action potential spreads from this point, so if you're having an action potential spreading to this point, so immediately it becomes, if the positive moves to this spot, this other part becomes negative, or it rests, because it does not hold this action potential for long. The movement is quite swift, unlike what we saw with the cardiac and the muscle cells, where with the calcium involved in the neuron propagation, we are likely to hold it a little bit. The action potential is likely to be held a little bit for long. So that's what happens with the movement of these ions. And eventually, this will move along the axon. So the initiation that we saw in the cell membrane, the cell body, spreading to the axons. So that means our impulse is spreading. So with the impulse spreading, we are saying that our place, or rather the cell, is being depolarized. Or you can say the wave of depolarization is spreading towards the axonal end. And remember, mathematically, we were able to say that depolarization, if you have a graphic representation, will be deemed as something that, let's say we are having our cell resting, and towards this side it will be on the action part. So while resting, the cell is somehow negatively charged. While this negativity reduces as you go up the ladder, approaching somewhere 0. So again, let's, for lack of no number, let's again use -61. This is the electrical voltage that is able to be calibrated. Of course, when we are having the number of sodium and potassium that we are able to see. So as potassium is being retained, as sodium enters, it means the cell will be becoming more positive, approaching 0. So approaching a point where there is no hole, or a point where there is no charge, onto the other opposite side. So moving towards this side, we will be saying that the cell is being depolarized. The depolarization, as per our text, representing an increased permeability to sodium. Of course, sodium needs to enter so that the positivity increase, and decreased permeability to potassium so that we retain a lot of it. So this is what generates the action potential. And it normally spreads across the along the membrane. So in that spreading manner, it represents a nerve impulse propagation or transfer. So this normally takes a very short or a rapid time. So the depolarization is very rapid, allowing conduction of impulse along the neurons within a few milliseconds. It passes from a point of stimulation to one direction only. So it's likely to move in one direction. Note that we are having some signal moving this other side. So the point of or the direction of the depolarization is normally a one direction thing. The same way we were able to see even with the GIT. why we were having our peristalsis moving from the oral, respecting the anal area. So that is a similarity when it comes to the propagation of these impulses. And of course, the propagation is because of the changes in permeability across the axonal lemma or across the membrane. And we've been able to say that while we are having we are shifting our positive, while our positive wave is spreading, so immediately our positive wave spread, so this cancels to become now the negative, it rests. And there's a point or a time where it needs to take some time before another action potential is possible. So this is regarded as the refractory period. So again, for pathological reasons, we should be able to note the importance of this. So this will mean that if a cell does not have a refractory period, or a neuron does not have a refractory period, it will mean that these are the people who will clinically experience seizure, convulsion, or epilepsy, because we are not likely to have a coordinated, let's say, a neuronal flow. So note this, when we'll be doing maybe some neuropharmacology, you'll be able to see how to modulate this refractory period, at least in attempt to control some form of convulsion. So when we're talking about the transfer or the conduction of this action potential, it can happen in two manners, depending on whether neuron is myelinated or not. So a myelinated neuron, it's likely to spread in what we call the saltatory manner, meaning that we are having a signal moving from this exposed area and then shifting up to this other point. So if you look, or rather if you are able to animate this so that it seems like maybe a continuous movement, so maybe demonstrate this, maybe animating itself, you are able to see as if it's jumping the myelin sheath. So we are told that this type of propagation is normally much quicker, much rapid, and this is why we were able to say that the preganglionic neurons, most of them are normally myelinated, and this could account for their quick neuronal relay or impulse transfer. So saltatory conduction that is linked to the myelinated. So we can have another one. Let's just go direct and skip the text. We can have a simple propagation where it will just be going straight. This normally happens in the unmyelinated, where we are just having a single, I don't want to use the term sheath, but let's use a single swan cell. So we are being told that this type of neuronal transfer faces a lot of resistance and is much slower. This is because the transfer of neurons to be much quicker depends on the area of the exposed part. So if you compare let's redraw our unmyelinated, that is, let's just be gross a little bit. So I will say that this is saltatory, while this one is simple propagation. So we've said for good movement, we need to be having some saltatory, and this is dependent on this exposed nodal area. So you can compare with this, that if you compare the movement, by the time you are reaching this other spot, this will be slower compared to this that has many surface area. not to assume that we are just having a movement like this, but rather these are just representing some form of wave. Some people might choose to say electromagnetic, having that there's some form of electricity. But what you just need to notice with the movement, if it was to move directly, it is faced with some form of, we can say, the insulation. So much is insulated. So here, minimal insulation, having it free, minimal. So maybe that's what we just need to note. So we are having two types of conduction, the saltatory and the simple propagation. We've said that the simple is likely to be slower, the saltatory is likely to be much faster, and then other parameter apart from unmyelinated or not, also the diameter of the neurons matter. A larger neuron is likely to be much faster than a smaller one. This is because this offers a good or a wide surface area for impulse transfer or propagation rather than the small. So the same thing that normally happens in electricity, for those who did electricity or physics. So a large wire is likely to transmit electric signal much quicker compared to the small one. This is because in terms of the surface area. So the same things apply to the nervous system, or rather to the neuron, having that it is also an electric unit. So eventually that is representing our electrical energy of the neural transmission. So eventually it will reach a synapse. So this is a point where axonal end, most of the time, we know we can have axonal, somatic, and many other, but let's just use a simplistic representation. Axonal end meets with the dendritic end. For this communication to be complete in terms of neuron propagation, there need some form of neurotransmitters. So neurotransmitters are chemical that acts as a relay tool for both for these neural impulses, of course, chemically. They need to be there. This is because they actually no physical contact between the two neurons, even though this one is subjected to debate, because when you examine the anatomically, it is not like a free space, but you can say that, to be more appropriate, you can say that there's no physical, and I'll add electrical contact between these two neurons. So maybe the space is maybe fibrous in nature that might not transmit electricity adequately. So because of this no electrical contact, there need to be some form of intervention, and this intervention is by the neurotransmitter. So the point at which the nerve's impulses passes from the presynaptic neuron to the postsynaptic neuron, this is called the synapse. So we are having two neurons joining. So just a simple presentation. So just like we were proposing about the ganglia stuff, of course the neurons that is giving the information will be regarded as the neuron. This one will be regarded as the or rather the post neuron. So, but when you respect the synaptic space, so this is the presynaptic neuron, and this is the postsynaptic neuron. So there are some form of neurotransmitter. There are forms of receptors that you'll anticipate in this area. And also good to know that in as much as this is also releasing neurotransmitters, it also has their receptors. We'll be able to see the exact role of the receptor. At its free end, the axonal on the presynaptic neuron break up into minute branches that terminate into small swelling called the synaptic knobs or terminals. So I think it is now the time to advance our axon a little bit. So it is not just a simple knob. We will be having also some form of subdivisions. I think this fair enough. So let's not overdo it. Swelling with some end swelling, having some knobs. Physiologically, we will be able to see why more than one synaptic knobs are very important. Later, we'll be able to see what happens when it is just one, what happens when it is just two. How does that corrupt the strength of neural transmission? But it is just good to note this at the moment. So these are what will be called, or rather called, the synaptic knobs. So these are in close proximity to the dendrites of the cell body of the neurons. So this meaning that it's a dendrite, another dendrite, another dendrite, another dendrite. Just draw as many as you can. Another dendrite. So that this becomes now our synaptic cleft. So this type of arrangement will be able to look at it when we'll be doing the neuronal mapping and circuits. But for now, let's just regard this place as a cleft. The synaptic knobs contain spherical membranes called the synaptic vesicles, which contains stored chemicals. So if we could just go with one buttons or with one synaptic knobs, this is the image that we'll get. So in a single knob, we are having vesicles. So these vesicles are important. This because they are acting as the storage unit for neurotransmitters. Just delaying within the knobs, they are just awaiting for the action potential. So when the action potential arrives, we'll be able to see what happens, possibly through these channels, called the calcium channels. So calciums will enter. So remember, these are voltage gated channel. That means they depend on some voltage to be open or to be closed. And remember, when the action potential is coming, it is coming with some sense of positivity. So if you take your graph, we are from -61, and again, for lack of no number, we give this at -40 or -40 to 20. So at this point, these electrical signal are very sufficient to allow the calcium channels to open. So when they open, they'll be able to act through the second messengers and stimulate the protein kinases, or very important enzymes that will allow this vesicle to phosphorylate and joins with the membrane of the synaptic knob. So when they join, they'll form a continuous membrane so that a neurotransmitter is eventually released via a process of exocytosis. So we are seeing how the electrical energy is being converted into the chemical. Voltage gated ion, able to use the second messengers, activate some enzymes, so that's already chemical by nature, and eventually releasing a neurotransmitter. So this could be acetylcholine, this could be adrenaline, this could be the gamma-aminobutyric acid, and many other, of course, including stuffs like also dopamine. And again, in a gross view, we've said that at the synaptic cleft, we anticipate an association of a neurotransmitter released by a presynaptic neuron to interact with the postsynaptic neuron. So ordinarily, there is usually some form of association. So the demonstration that we gave was with the axodendritic form of synapse. So that means an axon releasing a neurotransmitter interacting with the receptor of a dendritic, of a dendrite. So this association is referred to as the axodendritic. So this is a connection formed by the axon and dendrite. So this normally tends to serve an excitatory role. So excitatory role, that means we are having an impulse generated. When impulse generated at the synaptic point, it will be able to release neurotransmitter. Neurotransmitter will release and will interact with the receptor to cause another action potential or impulse generation. So that is what you'll say an excitatory. While we have the axosomatic, so that means we are having axon, an axon here. So this time round, instead of using a dendrite that has a receptor, it will interact direct with the receptor that is attached to the cell membrane of the cell body. So this is referred to as the axosomatic. So remember, we were able to say that cell body is also called the soma. So axosomatic kind of synapse. So this normally tends to be inhibitory. So inhibitory in a sense that impulse coming, there's some form of neurotransmitter release. Let's say let's talk of any inhibitory neurotransmitter. So let's say the gamma aminobutyric acid, the GABA. So interacting with this. So instead of generating another impulse or another action potential, it will cause these cells to be even more negative or allow them to rest even more. An example will be, there are some environment. Let's just create a scenario. Maybe you are in a class, or maybe you are traveling, listening to some music, or maybe listening to some boring lecture. You are getting some signals to your brain, but because the brain is interpreting them as somehow boring, it is releasing a neurotransmitter like the GABA, so that it causes an inhibitory signal. So with the inhibitory signals, these other parts become inactive, or rather rest. So grossly, it might be seem as if you look at the brain generally, it might fall into a sleep, because with the sleep, the activity of the neurons are reduced. Another good example will be things like pain. In our body, we have a natural painkiller system that will be able to see, or an opioid system. So when a pain modality is coming with some form of impulse, once at this area, it releases a neurotransmitter. So an example, let's talk of something like endorphin. So the endorphin, once it interacts with this, it is able to kill that signal. So the pain modality is not likely to be transmitted. So these are very important in the axosomatic connection. So let's remember this. So we have many, but we are just demonstrating a few. So we can also have the axonic axonic interaction. So that's mean you are having an axon. Ordinarily, where you'll anticipate a dendrite, there's another axon. But for it to be more practical, I normally tend to think of it as a neurotransmitter being released, because it will be difficult to get this kind of an association. But this is more of a physiological association. So at the axonal end, there's an impulse releasing a neurotransmitter. So let's talk of any neurotransmitter maybe that we know of, releasing things like norepinephrine. So this norepinephrine released as a neurotransmitter, remember we said that this also has some receptors. So the same neurotransmitter can act upon this receptor. So physiologically, I think you are seeing that association. An axon releasing a neurotransmitter to interact with an axon. So this normally serves a regulatory role. And this makes more sense with the example that we chose. Because when this releases norepinephrine, when this norepinephrine interacts with these receptors, it relays signal to this nerve to tell it that to reduce the rate of releasing the rate of release of this neurotransmitter. So it is almost having somehow an inhibitory role, but will not choose that word. We'll choose the word auto-regulatory. So it is able to regulate itself. Of course, when the norepinephrine will not be there in much quantity, it means that there'll be minimal interaction with these receptors. And if there's a minimal interaction in these receptors, it means that it will produce more. And when it produces more, it means we'll be having more available noradrenaline that can again interact with these receptors. And what will that do? It will send a negative feedback to cause the lesser release of the neurotransmitter. So let's just remember this, because we'll be applying this more often. I think we've talked of this, the mechanism by which action potential causes the neurotransmitter to be released. We've appreciated that there is some calcium channel gate, voltage gate that are basically relying on electrical signal to open. After opening, a lot of calcium will get into the cells, and after getting into the cells, they'll be able to cause some of the cellular secondary mechanisms by activating some enzyme that will see our synaptic vesicles interacting with the membrane, so that eventually the neurotransmitters are released via the exocytosis. And now to be a little bit more specific, of course, when a neurotransmitter is released, it ought to act by a receptor. So maybe we can explore the different types of receptors. We've been able to see the different types of association in terms of the different types of synapse. The axonal dendritic, which we've said is an excitatory. Let me revisit. The axosomatic and axoasonic that we've said is a regulatory, and the other one serves to be an inhibitory. So when a neurotransmitter is released, of course, it interacts with the receptor. So you consider receptor. Receptor has two parts. We have the binding component and the ionophore component. So it should be making sense. So let's just assume that this is a particular cell. let's make it a little bit thicker, so that the thicker part represents the cell membrane. So the parts that interacts with the neurotransmitter, so this is regarded as the binding component. So it normally protrudes outwards from the membrane into the synaptic cleft. So here, neurotransmitter is expected to bind with the presynaptic from the presynaptic terminal, and we having the ionophore component. This is this other component that passes through the cell membrane all the way up to the cytoplasmic area. So that is good to know. When we are covering the receptors as far as hormones were concerned, we talked of a major 3 types of receptors. For the neurons, the 2 types of receptors are the ionotropic, or rather the ion channel receptors, or the second messenger activator or receptor, or somebody might say thing like metabotropic. So with the ion channel receptor, that means that when a receptor is activated by a neurotransmitter, for us to experience either an inhibitory or an excitatory impulse generation, it will act so by allowing another channel to open, so that it either allows the influx of a negative for inhibitory reasons or a positive for excitatory reasons. So this stimulated, it directly stimulates these ion channels to open, and it is for that reason that it is regarded as an ion channel receptor. We having the second messenger receptor, as the word suggests, that when the receptor is activated by associating with the neurotransmitter, it will activate the second messenger system. So one of them that we know, cyclic, the cyclic adenosine monophosphate, the CAMP, or the cGMP, the guanosine monophosphate, and many other with the phosphate group will be able to see other types of second messenger. So when it is activated, it activates this particular system. So when these are activated, eventually it is able to give us a range of physiological activities. Some of them might end up opening an ion channel again, but considering the time in terms of activating these enzymes and these chemical secondary messengers to the opening of these ions, it will be a little bit slower, unlike this that seems to be direct. So with more explanation on the image, so consider this as a secondary, not secondary really, a second messenger receptor, that for a cell or for a neuron to experience a physiological activity, it depends on the activation of this not this, but rather second messenger system. So sometimes it can also be referred as a G-coupled receptor. So that is the secondary, the second messenger activated system. So when a neurotransmitter interacts with this receptor, what it does, through using either the alpha, the gamma, and the beta, the G protein, are able to modulate a lot of physiological activity. It might eventually opens the ion, opens the ion channel. It might activate an enzyme. It might activate transcription of specific genes, even though that is not as common. It might activate one or more other intracellular enzymes. Because these second messengers, you'll realize that these systems are a series of enzymes. Once this is activated, the X activate Y, Z, and the story continues from Z, A, Z. The story could be long. But one of the famous are the CAMP and the cGMP. So when these are activated, you can see what they can do. They can utilize the already available energy currency, the ATP and the GTP. So that is what we need to remember. Why are they important? This is because it will be able to tell us more when it comes to things to do with the speed, and things, of course, to do with the regulation of the same in a disease context, likely to be uncoordinated impulse relay. So when you go specifically into the ion channels, of course, there are two types. You can have some cation channels and ion channels. This is because we've said that whether you want to experience an excitatory or an inhibitory, it will depend on the type of ion that is activated. For example, neurotransmitters that we proposed a while back, such as the gamma-aminobutyric acid, to an extent endorphins, might act using an ion channel, meaning that when they are interacting with their receptors, it causes these ion channels to open. So ion channels, you can represent them as something like this. So so these are an ion channel. An ion channel allows negative ions to pass through. So one of the major negative ions charge an ion is normally the chloride. So you can guess the charge of these ions. So they are normally positively charged. So this is an ion channel, positively charged, so that when positive ions attempts to get in, they'll be repelled. So remember that it can easily attract the negative rather than the positive. So it is charged positive, but this one is an ion, or let's just note it to this. The opposite, a cation, allowing for positive to get in. Our major positive is normally the sodium, to an extent potassium, and also in some cases calcium. So of course, again, this will be negatively charged, the gates, with adequate activation, so that it adequately repel negative charge such as the chloride. So this, we attach it to cation channels. So this applies to the ion receptors. We've said that when ion receptors are activated, they'll open they'll open an ion channel. An ion channel, which one will be open? If it's a cation, will be having an excitatory, because remember, opening positive will allow the cell to be positively charged. Opening a negative will allow the cell to be negatively charged. So if the cell is negatively charged, it means that it will be resting. So unlike that which is positive, it means that you'll be in an action potential. Very important. So away from the neurons, maybe briefly with the neuroglia. So we'll just appreciate maybe their specific role that they do in the nervous system. So we said that the majority of the cells that we're likely to get in the nervous system are the glial cells, and we are having various types of glial cells that are doing other specific roles, but generally they can be deemed as a supportive cells that are not excitable. So action potential spreading, and we are having a glial cells not likely to go through it. they are just likely to use other neurons. So one of the glial cells are astrocytes, as the word suggests, star in shaped with fine branching processes. They lie in mucopolysaccharide ground substance. So what do? At the free end of some processes are small swelling called the foot processes. They are found in large number in adjacent to the blood vessels, with the foot processes forming a sleeve between them. So this means they can be very important in the architecture of the blood-brain barrier. So no need to draw, we are having the image over there. So other support, other function apart from the support, of course, structural, that is apart from the brain-blood barrier, there's the structural. It can give us the glycogen full reserve buffer. So remember, our neurons, we are majorly banking on some glucose. So these other cells can give us alternative for lack of word, let's say metabolic fuel, so that at least we can reconvert it back to glucose, feed our neurons with the glucose for their normal metabolism. So if you go deep into neurochemistry, those are the aspects that you can appreciate. So we've said that this is an astrocyte. So maybe back to our image, you extend this a little bit. So we are having the foot processes. So these foot processes are in close association with the blood capillaries. So if you take the cross section, so if there were some things or substance to flow from the capillary onto this particular space, you see the astrocyte is contributing by giving a barrier. So very, very important for the BBB. You're having a problem or a neuronal disease that is greatly getting rid of this, it means that the protective gear of the brain will be compromised, and as a result of that, some of the things that should not be able to find themselves into the CNS space will readily cross, even though not all areas of the brain are covered by the BBB. So I'll be interested if you have a finding of some of the specific areas of the brain that have these exemptions, so that we discuss in our next session. So these are the continual role: metabolic support, glucose sensing, transmitter uptake and release. So sometimes there are some neurotransmitters that might not be reabsorbed back by the presynaptic vesicles or membrane. So this can serve as an alternative. So exercise express plasma membrane transporters such as glutamate transporters for several neurotransmitters, including glutamate, ATP, GABA, metabolic and GABA. Metabolic support, we've said, they provide neurons with nutrients such as lactate. So remember the lactate. So lactate, we were able to say when you're having a lot of pyruvic acid accumulating, you're able to get the lactate. But we know alternatively where you can get the lactate. So we are having alternatives that can be later converted back into glucose for its use. Promoting myelin activity of the oligodendrocyte, nervous system repair. Upon injury, the nerve cells within the central nervous system astrocyte will fill up the space to form, fortunately, glial scar. Maybe one thing that we did not mention, compared to other cells, these are neural cells are cells that are slow in terms of regeneration. They are very slow in terms of regeneration. So cells like the astrocytes can come to their rescue by trying to repair, but unfortunately with a scar that will limit neuronal flow. Any sensory or motor modality will be greatly impacted, but at least there'll be a complete in terms of that physical link-up. So we also have oligodendrocyte. So much smaller than astrocyte. This found in cluster. They are normally deemed to have a physical supportive function. They are found adjacent to the long length of myelinated nerves. They form and maintain myelin-like swan cells in the central nervous system. So that is the oligodendrocyte. No, it should not be really on the central nervous system. So they form and maintain myelin-like cells in the peripheral nervous system. So maybe say you just cancel this, right? Peripheral. So in the CNS, you are having the swan cells. In the PNS, you are having the oligodendrocytes. We also have the ependymal cells. We'll be able to explore their role, very important, especially when we come to the secretion of the cerebral spinal fluid. So that means that there are some area, very important area in the brain, specifically in the ventricle, whose epithelium are lined by the ependymal cells that are able to allow some secretion of the cerebrospinal fluid. So should it be on this other side? Should be because we want to get into the central canal. So the opposite side, that is. So in this other area, there's some form of blood. So the exchange of not really an exchange, but we are getting nutrients, we are getting oxygens, we are getting a lot of substance from the blood into the CNS that will go to the central canal of the spinal cord, and also housing the meninges, because there is an association between the ventricles and the meninges, and also the central canal, or the to the again the spinal cord meninges, so that the brain is well backed with this fluid that will be serving various roles, as we'll be able to examine. We're having the microglia. So these are again very important, acting like some form of white blood cells, not really white blood cells, but some form of a defensive form of cells. This is because they are phagocytic in nature, so they assist in removing microbes, damaged tissues, in area of inflammation and cell destructions. Very important. So let's end this session at this point. In our next episode, we'll start with the central nervous system, of course with major emphasis on the brain and the spinal cord. And remember, this is just mostly before we get now into the real content of some of the physiological, or rather the electrical mapping in the nervous system. So just read more on this, and then we'll pick it from this point.